Hello, I'm Prof. Grivas. I'm a medical oncologist at the Seattle Cancer Care Alliance. I'm an associate professor and the clinical director of the genitourinary cancers program, and I'm also an associate member of the Fred Hutchinson Cancer Research Center. This is a single-arm phase 2 trial of this novel and interesting molecule called bintrafusp alfa. This is a very innovative compound that inhibits PD-L1 and TGF-beta concurrently. So it aims to inhibit the role of TGF-beta, which can mediate immunosuppression. And this molecule is given as single agent in patients with platinum refractory advanced urothelial cancer, so patients had progression on prior platinum-based chemotherapy, but have not received immunotherapy yet. So it's a purely second-line study in advanced urothelial cancer in patients who have progression resistance to platinum-based chemotherapy, but have not received immunotherapy yet. Absolutely. So there has been a lot of data from our group, as well as Dr. Galski's group, Dr. Marianne Hassan, and others, suggesting that the tumor microenvironment, the tumor tissue stroma, may be very relevant in modulating immune system response, especially when you think about checkpoint inhibitors against PD-L1, PD-1 pathway. And particularly TGF-beta has come a very important molecule has been identified as a major mechanism of resistance to immune checkpoint inhibitors based on these translational studies. And we have shown before, and we have presented in meetings, that a signature that is driven by TGF-beta in the tumor microenvironment may suppress the immune system and may, I would say, negatively impact, may impair response to immune checkpoint inhibitors. So the concept here is if you inhibit this potential mechanism of resistance, TGF-beta and PD-L1 at the same time, could you potentially achieve higher responses compared to PD-L1 inhibition alone? And based on that notion, this molecule was developed, and in this phase 2 trial, the primary endpoint is overall response rates to evaluate whether this concurrent inhibition of PD-L1 and TGF-beta can result in higher response rates compared to historical control with PD-L1 inhibitor alone. There have been data regarding this agent in other prior studies, and this has shown that the molecule appears to be well tolerated, and the safety profile appears manageable. And overall, based on this early safety data, this trial actually was conducted. And as I mentioned before, this particular study looks at a particular dose of this agent, the bintrafusp alfa. Just to clarify the point, this is a phase 1b study, not a phase 2. Phase 1b, just to evaluate the efficacy and the safety further of this particular molecule. So the goal of this study, it's a phase 1b, as I mentioned, is to evaluate both efficacy and further the safety in patients with metastatic platinum refractory urothelial carcinoma. So that's a big question. That's a great question, whether the concurrent combination of PD-L1 and TGF-beta inhibition can result in higher response rates and better outcomes compared to PD-L1 inhibition alone. I think that's the promise, and that's the intent of this study, to evaluate further, as I mentioned, the efficacy and safety. As I mentioned before, the inhibition of the TGF-beta R2, which is, again, mediating resistance, is a very, I would say, novel and promising idea. However, we need to further evaluate the safety that was shown in phase 1 trials in this particular phase 1b study, and also the efficacy in this particular patient population. So I think it's too early to tell. I think the mechanism of action is very compelling, and I think it's definitely a very exciting study, but we have to see exactly what happens with the results and further evaluate the fate of this molecule, particularly in platinum refractory advanced urothelial cancer. And if we see a promising response rate, the question will be whether we move that a molecule potentially earlier in the disease space. We know that avelumab switch maintenance is now standard of care after response or stable disease to induction platinum-based chemotherapy. But before we do a large maintenance study, we need to have a proof of principle efficacy phase 1b study, and that's why we do this particular one now.